How is it that a microscopic organism can survive everywhere from tropical lakes to boiling hot springs, parched deserts, hypersaline environments, and even freezing habitats, and not just survive, but sometimes completely transform an entire aquatic ecosystem? It's a massive, profound scientific question, but it's the actual, everyday reality for one of Earth's most incredibly adaptable life forms.
Welcome to this explainer, diving deep into Dr. Indunal Senanayake's lecture on the occurrence of cyanobacteria. Today, we're going to establish a fundamental truth right out of the gate. Cyanobacteria are not just simple, microscopic organisms. They are master ecological engineers. They're vital primary producers, incredibly fierce microbial competitors, and in certain cases, they produce some genuinely powerful toxins.
Okay, let's look at our roadmap for today. 1. Meet the ecological engineers. 2. Survival across extreme environments. 3. Taxonomy and structural diversity. 4. Ecological roles and interactions. And 5. The dark side, cyanotoxins.
Let's kick things off with Section 1. Meet the ecological engineers and why we need to redefine blue-green algae.
Now, you might have historically heard these organisms misidentified as blue-green algae, but here's the thing, they aren't algae at all. They're actually highly diverse prokaryotic microorganisms. When we look at their 3D architecture, this distinction becomes super clear. Because they're prokaryotes, they completely lack a true nucleus. Their DNA ring just floats freely inside. Yet, hidden right beneath their peptidoglycan layer and protective slime coat, they have highly advanced plant-like photosynthetic machinery. Specifically, we're talking about those green thylakoid rings. This elegant biological contradiction, a prokaryote doing complex photosynthesis, is really the foundation of their global success.
Moving right along to Section 2. Survival across extreme environments, a global distribution.
At their core, cyanobacteria really just rely on the basics: water, carbon dioxide, light, and inorganic substances. But their real secret superpower comes down to a simple ecological equation. Profound environmental tolerance plus physiological adaptation equals a massive, broad ecological distribution. This adaptability is what allows them to journey way beyond your standard freshwater and marine systems. I mean, they literally dominate extreme habitats. We're talking boiling hot springs, hypersaline pools, freezing glacial environments, and bone-dry arid deserts. They thrive in the exact extreme spots where true microalgae just completely fail to survive.
All right, let's zoom in a bit for Section 3, Taxonomy and Structural Diversity, the 4 orders.
To really understand how they pull off this insane adaptability, we have to look at their structural complexity. What we have here is our taxonomic blueprint for cyanobacterial diversity, organizing roughly 150 identified genera based on their cellular architecture and filament organization. We're going to build our understanding from the ground up today, scaling from the most basic, simple unicells all the way up to complex, true branching structures.
We begin with the order Chroococcales. Genera in this group, like Microcystis, Merismopia, Gloocapsa, and Aphanocapsa, feature spherical, ovoid, or cylindrical cells. Now, right after division, they might just exist as singular cells. But here's the cool part. As they mature, they aggregate into these highly complex colonies, all locked together and secured within a really resilient slimy matrix. It's essentially a microscopic metropolis wrapped in protective slime. And this brilliantly illustrates our next jump in complexity, scaling upward to the order Oscillatoriales. Here, the single cells form elongated multicellular strands. These are known as uniseriate filaments or trichomes. We see this exact structure in Oscillatoria, Lyngbia, Formidium, Microorganisms, and Planktothrix. But crucially, while they form these really long chains, they lack true branching, and they don't possess any specialized cells.
That changes when we hit the Chroococcales order, which introduces profound heterogeneous cellular composition. Basically, alongside their standard vegetative cells, they start developing some incredibly specialized tools. First, heterocysts. These are specialized cells with a thick, hyaline protoplast engineered entirely for fixing nitrogen. Second, they form aconids. Think of aconids as heavily fortified, thick-walled survival pods that they use to ride out hostile environmental conditions. This advanced cellular division of labor is a hallmark of genera like Anabaena, Nostoc, Cylindrospermopsis, and Calothrix.
Let's move to and see how this builds up to the absolute apex of cyanobacterial complexity, the stigonematals. Just like Nostocalis, they utilize both those specialized heterocysts and echinotes. But the massive evolutionary leap here is in their filament organization.
While Nostocalis are strictly unbranched or exhibit false branching, Stiginonematals genera, like Stygonema and Mastigocladus, actually form uni- or multiseriate trichomes capable of true branching.
So the crucial point is really seeing how this architectural evolution stacks up. From the simple, slime-bound colonies of Chroococcales, we step up to the unbranched filaments of Oscillatoriales. Then, Nostocalis introduces those powerful specialized cells for nitrogen fixation and survival. And finally, Stiginometales takes all those specialized tools and organizes them into complex, true branching structures. This increasing structural mastery is exactly what drives their global dominance.
Next up, Section 4, Ecological Roles and Interactions within the microbial community.
Because they operate as aerobic photoautotrophs, cyanobacteria deploy some genuinely incredible survival tactics in the wild. For example, they can actually modulate their own buoyancy using internal gas vacuoles. This lets them float up or sink down to either escape or absorb UV radiation. Certain species can also create massive microbial mats that tolerate very low oxygen. And from an ecological standpoint, they are absolutely indispensable for nutrient cycling, driving both nitrification and denitrification. In fact, their nitrogen-fixing capabilities are so potent that symbiotic systems, like the azolifern, are heavily utilized globally as natural agricultural biofertilizers for rice production.
But of course, there is a flip side to all this power. Section 5, the dark side, cyanotoxins and competition.
When a master ecological engineer overdominates its environment, the consequences can be incredibly severe. Massive cyanobacterial blooms can synthesize natural cyanotoxins. We're talking about a diverse chemical arsenal that can cause hepatotoxic liver damage, neurotoxic failures, dermotoxic skin reactions, and even the broad inhibition of protein synthesis. Curiously enough, these toxins are often way more hazardous to terrestrial mammals that happen to drink from the water source than they are to the surrounding aquatic life. Though, fascinatingly, some specific aquatic bacteria have actually evolved the ability to naturally degrade these toxic compounds.
And beyond just poisoning mammals, cyanobacteria engage in fierce, microscopic chemical warfare with their neighbors. When a dominant bloom takes over, competing phytoplankton practically vanish. How? Cyanobacteria achieve this by releasing targeted allopathic compounds directly into the water. For instance, a species like Microcystis aeruginosa will aggressively inhibit the growth of diatoms and other algae, while Oscillatoria directly suppresses the reproduction of key competitors like Cryptomonas. They literally chemically clear the field for themselves.
Because they are so globally dominant, it's absolutely crucial that we apply rigorous, localized scientific scrutiny. The source material for our explainer really hammers home the critical nature of regional investigations, specifically highlighting Professor Pothamala's extensive research on cyanobacteria in Sri Lanka. Identifying the exact regional blooms, understanding specific local genera, and tracking their distinct toxin profiles isn't just an academic exercise. It is paramount for safeguarding public water systems, health, and local agriculture. So from totally redefining photosynthesis in prokaryotes to engineering vast microbial communities to surviving the absolute harshest climates on Earth, cyanobacteria are undeniable masters of their environment. Which leaves us with a final, rather pressing question. If cyanobacteria are this perfectly adapted to engineer ecosystems and toxically outcompete other forms of life, how will their dominance reshape our planet's aquatic systems as global environmental conditions inevitably change? It's a striking reality, and one that absolutely demands our utmost scientific attention.